All right. Well, thank you, Tom, and thank you to the organizers for putting this together. And yep, I'll be talking about amphibians and their parasites. And first off, I just want to acknowledge a number of folks and organizations that contributed to this work. So in particular, a number of undergraduate as well as high school students actually participate in the research I'm going to be talking about. Some of the work I did was performed at the Ammonium Field Station. And we've had support for some of the student involvement from organizations like the National Great Rivers Research and Education Center and the Bradley BEST program. So it's certainly been a collaborative effort, and I appreciate all the organizations and people who've contributed. So I do research on parasites, and in our current environment, I think everyone appreciates that pathogens and parasites can have important consequences for populations. And there have been a number of documented cases in wildlife. So white nose syndrome in bats, having devastating population effects, the effect of the sea lamprey on lake trout in the Great Lakes, effects of pathogens on zooplankton populations that control their populations, parasites of amphibians causing limb deformities, and sarcoptic mange and wolves affecting populations in Yellowstone. So a number of cases that you may be familiar with. And if you're studying host-parasite interactions, though, oftentimes researchers will focus on this interaction in isolation, but it doesn't happen in isolation. It's actually embedded within a set of complex systems. For instance, the host is typically going to be embedded within a food web, and in the simplest case, it has some food resource that it uses and maybe a potential competitor. There also may be predators or other species that it's interacting with. And then the parasite also may be embedded within a complex system, like a complex life cycle. For instance, your focal host may just be an intermediate host in a complex life cycle that gets infected by a free-living parasite stage, but that parasite also has other hosts involved in its life cycle, and those hosts may be affected by a number of different ecological factors. Adding to that complexity is human impacts, and a number of systems, in particular freshwater systems, are undergoing one major shift as a result of changing nutrient regimes, in particular due to eutrophication. And so you could imagine that these changing nutrient regimes would have consequences for a complex system like a food web or a complex life cycle of a parasite, and it may be through multiple different mechanisms. So when you have fertilizer inputs that via runoff make their way into a wetland or another aquatic system where you have an aquatic host, there can be potential impacts, for instance, on other hosts in the life cycle. There could be potential negative effects of the fertilizer, potential toxic effects of fertilizer inputs directly on the host, as well as potential changes in food resource availability, in particular, if that host eats algae, for instance, that might increase algal productivity or change it in some other respect. So a number of different mechanisms, and it does make a challenge for trying to predict what's going to happen when you have these changing nutrient regimes in aquatic systems that have these types of interactions going on within them. So that motivates our overarching objective for what my lab group has been doing is trying to understand the mechanisms by which these changing nutrient levels influence host-parasite interactions. And the system that we study this in is, for our host, we focus on amphibians, which are beautiful, but also an important ecological component of many ecosystems. And they provide an important link between aquatic and terrestrial ecosystems. And from a conservation perspective, they're important. They've been experiencing recent global declines, in part due to factors such as parasitism, as well as due to pollution. And increasingly, they've been used also just as a model for understanding food web interactions, including host-parasite interactions. So there's a decent amount of information that we can build on in our research. And for performing experiments, as well as performing field-based research, they're a useful model from that perspective as well. And then the parasites we focus on are trematodes, which are a very common group of parasites and quite diverse. Within a particular aquatic habitat, you may find a dozen or more different trematode species that can have pathogenic effects on animals living in that, living in that habitat, including amphibians, as well as other wildlife that use that habitat. And trematodes typically have complex life cycles involving multiple hosts. So the first host is always going to be a mollusk. And then amphibians often will be a second intermediate host if they're involved in the life cycle, with another vertebrate being the definitive host. And their distributions can be influenced by a number of different ecological factors, including nutrients. And there's been studies on nutrient effects in amphibian host parasite interactions with trematodes, as well as other factors. Again, there's a long list of other potential factors that can influence them as well. So the group of parasites, the group of trematodes that we focus on, parasitized amphibians, typically larval amphibians, are the second intermediate host for the parasites that we're focused on. And for several of the groups, when those tadpoles, those larval frogs, get consumed by some vertebrate like a waterfowl or mammal, that allows complete development to the adult stage. There's sexual reproduction within the gut of the definitive host. And then eggs will typically pass out with the waste, hatch, and then a free swimming stage will infect the first intermediate host, which is that mollusk host. And for our purposes, we focus on snail hosts. And then a free swimming stage will leave the snail host, so there can actually be asexual reproduction within the snail host, and then a free swimming stage will leave, and that's what either burrows into the skin or enters through an orifice of the tadpole to infect it, depending on, again, what type of trematode you're talking about. And we expect that there are potential effects of nutrients on this system through multiple different mechanisms. Probably the one that's been best studied is potential effects on the snail host, where more nutrients leads to more algal productivity, that leads to more snail productivity, and also potentially more infection in the snail host. And so we tried to look at this locally by doing surveys of wetlands in Illinois. And I'm going to focus just on a subset of the wetlands we sampled that we started sampling last summer. That was on the series of wetlands that Krista was just talking about on Franklin Farm, where we know there's a gradient in nutrient concentrations in those wetlands, so we thought would be useful for trying to examine potential nutrient effects. So we collected 2 snail species that are hosts for trematodes, and then looked for a diverse set of different trematode species. I'm not going to talk too much about different types of trematodes in this talk, but I can get into that in the questions if folks would like. So Krista already showed a picture of where the wetlands are. So I was only able to actually collect snails from the full series for the Gully series and the West series. So there's those are 2 of the 3 series that are present on Franklin Farm, the farm that Krista introduced in her talk. And both of these series provide gradients in nutrient levels. And the reason that gradient occurs is because the tile actually just drains into the first wetland in each series, as Krista explained, and then nutrient loading will consequently decrease as you move from the first to the third wetland in the series. And so in our survey, we collected 2 species of aquatic snails. Within each wetland, we estimated the snail density, as well as when we were able to collect enough snails to estimate prevalence, we also examined trematode prevalence. And we had generally the prediction that more nutrients would lead to higher snail densities, as well as higher prevalence. So to assess prevalence, to actually screen how infected the snail first intermediate hosts are, you can basically take individual snails, you put them into individual cups, and then you put them under a light and examine them after at least a few hours. And the free swimming stage will actually leave the snail. The light induces the parasite to leave the snail, and then you can assess its infection status. And then we can examine them under a microscope to say whether or not it was infected and with what type of trematode it was infected with. So first off, just looking at snail density across the different wetland series, so West and Gully series, where we measure density as catch per unit effort. We saw a lot of variability generally, I'll say. So for plantar bella, at least on some occasions, we saw evidence of a trend in the Gully series. In the West series, not much of a trend. For Physa, the other species we looked at, we didn't see much of a trend in the West series. We did see evidence of a trend in the Gully series, suggesting potential declines in density as you move from the high nutrient to the low nutrient ponds in each series or wetlands in each series. But not a obvious pattern that's consistent across all species and series. If you look at actual infection prevalence, the percent of infected snails. Again, so in the top we have for the first host species, the bottom for the second host species. You're looking at the West, as well as, we only had data in June for the Gully Series because we didn't get enough snails in the Gully Series in July. At least for the West Series in June, we did see a negative trend of lower prevalence as you moved through the series. And we saw that for all FISA species. We didn't see any trend, though, for plant rubella in the West Series in July or June. But we never saw the opposite pattern we predicted. So at least preliminarily, we can say there's some evidence that there may be the predicted trends, a potential effect of nutrients, or some of the other factors that could be going on as you change from one pond to the next. But at least it's consistent with what we would expect, although it is clearly quite variable. So we have some evidence potentially for this mechanism that higher nutrients is influencing snail populations, but there are other potential mechanisms that nutrients could be influencing this interaction as well. For instance, nutrients are also going to be affecting algal growth in the ponds that could influence the amphibians, both by influencing the amount of algae as well as what the composition of algae is. So we've actually done some experiments in the laboratory looking at how food resource quality, which you would expect to vary with algal amount and composition, what changes in that would how that would influence a parasite infection. So I did some work previously a few years ago looking at how food quantity influences infection loads, where generally I found, basically I fed tadpoles 2 different food resource levels, and then I measured infection when they were exposed to an infected snail that was continuously producing parasites. And I found generally that ones that were fed lower food levels tended to be less infected than ones fed higher food levels. The reason I think that this result occurred is basically when there's more food resources, tadpoles grow more and larger individuals are larger targets for the parasite. And so they end up getting more infected. And we've seen that in some other experiments as well, that body size is associated with more infection. More recently, a student in my lab, Kiernan Robinson, has looked at how food quality as opposed to food quantity influences parasite infection, where he's done some experiments manipulating the ratio of protein to carbohydrates and done some controlled exposures of tadpoles to parasites. And generally what he's shown is that as you increase the amount of carbohydrates relative to protein, you tend to see higher infection susceptibility in tadpoles. So food quality also appears to have an influence on parasite risk as well. And then just briefly, I want to talk about another potential mechanism, which is potential direct effects of fertilizer or other indirect effects of fertilizer on amphibians that we want to address experimentally. So we did some mesocosm experiments at the field station, looking at, in particular, we focused on ammonium nitrate impact. So we added ammonium nitrate, and then we also manipulated the presence or absence of parasites by including infected or uninfected snails. Set these basically to mimic natural ponds, where we added zooplankton, phytoplankton, leaf litter as substrate, and then we added tadpoles to each tank to see what the combined effects are of added nutrients as well as added the exposure to parasites or not. And what we found was somewhat surprising in that we didn't see much of an effect of the parasites, but the nitrate had a very strong negative effect on survivorship of the tadpoles. Basically, tanks we added nitrogen to had very low survival overall. Now, there's been some evidence of nitrate toxicity. We did do experiments in the lab where basically the concentrations we used in our mesocosm experiment, we didn't see any effect in the lab. In the field, we saw much reduced survival of tadpoles. There is some evidence that nitrate in combination with other factors like UV exposure, which would happen in large tanks in the field that wouldn't happen in our laboratory experiments, that combined effect may have amplified the effect of nitrate. But still, we were quite surprised at how strong the negative effect of nitrate was on survival. Interestingly, we did also see an effect of nitrate on parasite infection, where actually there was lower infection when nitrate was present compared to tanks where there was parasites but no nitrate added, or there was still nitrate present, but much lower nitrate. We didn't add the additional nitrate on top of that, suggesting that at least there's some weak indirect effect of nitrate that is reducing parasite exposure, potentially because the nitrate is also negatively affecting the parasite or the snail that's producing the parasite, although clearly that effect is largely outweighed by the negative effect on survivorship of the nitrate exposure. So just to quickly draw some general conclusions, at least based on our survey results, we see potential effects of nutrients, although we can't completely rule out other factors, and the data are quite noisy. So we do want to continue this survey in additional years to survey these ponds. And also, actually, we have amphibians hosts that we've collected from these ponds that we're going to assess infection levels to see what the consequences of changing snail density and prevalence are for amphibian infection. We also have evidence, at least from our lab study, that changing food resources, both quantity and quality, may influence parasite susceptibility. And then, in general, it appears that ammonium nitrate may have some pretty negative effects for both the host and the parasite in the system. So that's something we want to follow up on and try to integrate how these different mechanisms are playing off each other to actually influence populations in the field. And we think this is going to be pretty important to do, given the fact that these types of systems are undergoing changing nutrient regimes and the threats that are facing wildlife like amphibians, including factors like disease. So with that, thank you, and I'm happy to take any questions.
Thank you, John. Doug, we do have about 2 minutes for questions, if you have any. You're muted, Doug.
I think he covered everything so well. No questions so far. So I might ask, John, how do you see the predicted climate change in this area impacting those relationships, if at all?
Oh, I think it could have a huge impact. Yeah, so I mean, already the system is very complex, and then you add an additional factor on top of it. But I mean, one thing is, the occurrence of storms is going to influence nutrient inputs. So that's one thing. As more runoff could lead to more nutrient inputs into ponds. And then warmer temperatures or changes in temperatures are going to influence how well the hosts are able to tolerate the parasites. And there's been studies showing that basically you add climate change on top of parasite exposure that stresses out tadpoles and leads to amplify negative effects of the parasites. And so I think it adds more problems on top of it.
Here, I've got another one. Was the level of ammonium nitrate added in the lab or field experiments? Was it a high, low, or moderate amount?
Yeah, so it was high. It was on the high end. It was certainly within the realm of what occurs, but we wanted to I guess, first off, explore whether or not we'd see any effect at all, because other people had seen it for some species, but not others, an effect of ammonium nitrate on amphibians. So we didn't know if we'd detect it. This past summer, I tried to follow it up by actually doing more of a gradient approach where we went from low to high and with some intermediate approaches. But those data, well, we had some problems with animals colonizing our mesocosm tanks. So those data are a bit too messy to really draw many strong conclusions from. So I think that's something I'm going to have to follow up more on in the future.